Good evening, everyone. Today is the fifth lecture which is a part of our talk series Bio-Illumination, which has been organized by our Department of Biological Sciences, Alia University. And before we start with the proceedings, some common instructions for all the participants who are joining us. First, please keep your microphones muted and don't start any presentation at all from your side. Second, all the questions that you need to ask, please put it in the chat box and they will all be discussed after the talk has been taken over. So don't ask questions through verbal, just message in the chat box and all of them will be taken up after the talk is over. So once again, on behalf of the organization committee, myself, Dr. Safdar Ali, convener for Bio-Illumination, welcome all the participants to this talk. We have with us Dr. Kazi Mirajul Haque, our honorable speaker, our head of the department, Dr. Masroor Alam, different faculty members and participants who are joining us from different parts of the country. So I welcome you all. Without further ado, I would like to call upon our head of the department, Dr. Masroor Alam, to say a few words to inaugurate the proceedings. Over to you, Dr. Alam.
Okay, thank you, Dr. Ali. Good evening, everyone. Today's speaker, Dr. Kazi Mirajul Haque, and other esteemed colleagues and other participants, scholars, and students. I hope that all of you are doing well in this COVID-19 pandemic. First of all, I want to thank Dr. Haque for giving us his time for this lecture, despite of his busy schedule there. And I also want to thank all our participants for attending our Bio-Illumination talk series regularly, especially those who are from other institutes and attending each of the lectures we are organizing. Some of the participants are very consistently attending all of our lectures. So a heartfelt gratitude to all of you for making our efforts so successful. Today is our fifth lecture in this series, and I should say first one, which is directly related to the COVID-19, and that lecture will be given by Dr. Haque, who is from University of Maryland, USA, who will deliver today's lecture on the topic flooding of pulmonary alveoli in COVID-19, the pathophysiological aspects of that. I hope that in this ongoing period of COVID-19, this is going to be a very pertinent talk, and we will gain some interesting knowledge COVID-19 pathogenesis. So not wasting much time, on behalf of the Department of Biological Sciences of Alia University, I heartily welcome you all in today's lecture. So thank you all, and let's proceed to the presentation.
Good evening to all of you present here. I am Dr. Sheikh Kobita, Assistant Professor, Department of Biological Sciences, Alia University. Welcome you all. Today we have a very relevant area to discuss on. The topic has already mentioned our head of the department, that is flooding of pulmonary alveoli in COVID-19, the pathophysiology. COVID-19 crisis is an unprecedented one in the history of mankind, and I believe that today's topic will be very relevant from this perspective. So I feel very privileged to introduce today's speaker, Kazi Mirajul Haque. Currently, he is a research associate, that is a faculty level equivalent position in the Department of Physiology, University of Maryland, School of Medicine. Dr. Haque did his PhD from Bardon University and BSc from Bardon University and MSc from Vidyasagar University. He completed his PhD from Jadavpur University. Hello, is there any problem?
Okay, ma'am, proceed with your introduction.
Okay. He completed his PhD from Jadavpur University from in the year of 2003 in the area of gastrointestinal physiology. He was a research fellow ICMR Research Fellow of National Institute of Cholera and Enteric Diseases, Kolkata. He was also postdoctoral fellow of Department of Digestive Diseases, Yale University School of Medicine, and Johns Hopkins University, Baltimore, USA. From 2009 to 11, he was the instructor of medicine in the Department of Medicine, Johns Hopkins University, USA. 2010 to 16, he was an assistant professor equivalent position in pathophysiology division, National Institute of Cholera and Enteric Diseases, Kolkata. He received a total 14 prestigious national and international research award. I'm not going to the details of that. He has a considerable number of research publications. He published his work more than 25 reputed peer-reviewed journals and book chapters. But I must mention recently he has published his work in Nature publication. Congratulations, sir. He is member of many research organizations like American Gastroenterological Association and many more. He is reviewer of 14 research journals and journal editor and editorial board member of many journals. Dr. Haque has undertaken various research projects, and presently he is a co-PI of a research project that is on COVID from NIH. And the goal of that is of more than 1 crore, and the goal of this proposal is the identification of altered ion transport in airway epithelia that has a critical implication in the physiology of COVID-19, and that is funded by NIH India. He is a consultant of a research grant. Presently, he is consultant of a research grant funded by Cystic Fibrosis Foundation, that is also of more than 1 crore. The targeting kinase signaling to treat CF intestinal diseases. And he successfully completed several projects from Government of India. He was principal investigator from ICMR research project, DBT research project, Rama Lingoswamy grant, Department of Biotechnology, Ministry of Science and Technology, Government of India. And I must add that he supervised many number of PhD students. So I want to express that the Department of Biological Science is indeed honored to have you here, sir. So with these few words, I would like to switch this virtual space to today's esteemed speaker, Dr. Kazi Mirajul Haque, to start today's session. Sir, please.
Okay, thank you, Kavita, for the nice and brief introduction. I'm really honored, and this is my pleasure, to be with you to present. I think it may be helpful regarding the COVID-19 on the pathophysiological aspect. Let me bring up my presentation. Let's see whether I can do it or not. How can I share my presentation? I don't know. I am very much familiar with the Zoom. I'm very new to Google Meet. Okay, let's see.
So there will be an option present now at the bottom of the screen.
Yeah, I'm clicking on that, and then I'm getting 3 options. Your entire screen, a window.
Yes, select entire screen.
Yeah, I did, and then.
Okay, उसके नीचे okay होगा sir, उसको okay करने के लिए.
No, it's saying share and cancel. The share button is not
share
active. It's not active, so I cannot do anything.
So kindly select the screen of your laptop, then select accept.
Let's see. No. Oh, okay, hold on. Now it is saying share. Okay, hold on.
Yes, sir, it has started.
No,
So now you just need to open your file. Minimize the meet window. Yeah. And then open the file that you need to present.
Yeah, that's the one.
Yeah. Now
Are
just go
you
for
seeing my
present.
presentation now?
Yes, sir. Just go to the full
Okay,
screen view.
that's good. That's good.
Yes,
Thank
sir.
you. Okay.
And you can just hide over there. What is showing now? Stop sharing and hide your in that small
Okay.
tab that is being over there.
Now, can you see my pointer also?
Yes, sir. Yes.
Okay, thank you very much. Thanks, Kavita and other member of the Bio-Illumination of the earlier university for inviting me to present on this topic. And thanks Kavita and other people also for nice introduction about me. Yes, I am a practicing scientist in the field of epithelial-linked disease that covers disease related to gastrointestinal, disease related to renal epithelia, and disease related to respiratory disorders, including currently I am interested on the pathophysiological aspect of COVID-19. So the title itself is probably giving you a sense what I'm going to talk about is the flooding of pulmonary alveoli in COVID-19. Specifically, I'll be talking how SARS-CoV-2 infection causes the lung to become inflamed, and the consequences of this inflammation leads to fill alveoli with fluid that COVID-19 patients struggle to provide enough oxygen to the body, which is termed as ARDS, or acute respiratory distress syndrome. So that I'll be talking about today. Let me bring the next slide before moving to the actual presentation. So on your right side, demonstrate the number of COVID-19 cases reported weekly by WHO, which is the World Health Body, on the global date, showing the global date of 30th December 2019 through 20th September 2020, which is a very recent one. And if you see from this figure, you'll see that globally, about 32 million confirmed COVID-19 cases, with almost 1 million deaths have been reported. Almost 1 million deaths have been reported to WHO as of 20th September 2020. And from 14th to 20th September, there were almost 2 million new cases of COVID-19, which represents a 6% increment compared to the previous week. If you compare the last bar with this previous one, you'll see there is increment. What does it mean is that still COVID-19 positive cases increases. There is no way that could tell us a sign of reduction of infection. With the exception of the African region, an increase in the weekly case incidence was reported across all WHO region in the last 7 days, as you can see from here. America, Southeast Asia, Europe, Eastern Mediterranean, Western Pacific, Africa, all, everywhere, still, the positive number of positive cases increases. Overall, the region of the America continues to carry the highest burden of COVID-19 globally, accounting for over 38% of all new cases reported in the past 7 days. From the last night report, 207,000 deaths happened here in the US. So still is a big threat across the world, specifically in USA, since there is no vaccine available against COVID-19. Let me bring next slide to share what is happening in our region, Southeast Asia. On the right-hand figure basically demonstrates the number of COVID-19 cases and deaths reported weekly to the WHO for Southeast region as of 20th September. The Southeast Asian region is the second most after USA. Is the affected region and currently account for 35% and 25% of cases and death respectively. The countries reporting the highest number of new cases from the Southeast Asia continue to be India, Indonesia, and Bangladesh. And the countries reporting the highest number of new deaths per million population include India, Maldives, with 6 and 4 deaths per million population respectively. Seems like, very few, 6 and 4, but because of the high population in our region, that's why you can see it is a low number here, okay? Now, let me give you some background about the COVID-19. So many of you know, in December 2019, the outbreak of novel coronavirus disease in China spread worldwide to almost 140 other countries, becoming an emergency of major international concern or health threat. So right now, the total number of positive cases exactly from the last night report is almost like more than 32 million. Really is a big threat to our civilization, human civilization. SARS-CoV-2, which is the causative agent for the COVID-19 epidemic, which causes the severe respiratory illness, similar to severe acute respiratory syndrome coronavirus. Phylogenetic analysis shows that SARS-CoV-2 is a new member of the coronavirus family, but is distinct from other two coronavirus called SARS-CoV and MERS-CoV, which causes the pandemic in 2002 and 12. If you look at the phylogenetic analysis, so there is a identity, almost like 79% with SARS-CoV and 50% with MERS-CoV. So basically, the SARS-CoV-2 is a new member and is a new virus of this family that causes this epidemic. And the transmission generally happens from human to human via droplets, contaminated hands or surfaces, has been described with incubation times of 2 to 14 days. And generally, early diagnosis, quarantine, and supportive treatment so far are essential to cure patients right now. Now, let me talk about the coronavirus. I'm very sure most of you know, using the Google search, the coronavirus are a group of RNA virus that causes disease in mammals and birds. And there are many different kinds. Coronavirus is basically named for the crown-like spike, as you can see here in a red color. So corona meaning is a crown. And it is a positive sense RNA virus that belongs to coronavirus subfamily. And this spike protein is really very important. And most of the mutation happen in this spike protein that make them so dangerous, so deadly. And the frequency of the mutation happen so frequently in this protein, in the spike protein. So this virus basically processes a single-stranded RNA genome wrapped in nucleocapsid, or N protein, and 3 major surface protein, as mentioned here, like M protein, envelope protein, and spike protein. So very important word is the spike protein that helps interact with the human cells, respiratory cells, or pneumocytes. It's called pneumocytes, more specifically. So until December 2019, only 6 different coronaviruses were known to infect humans, and 4 of these are HCOV NL63, HCOV 229E, HCOV OC43, and HKE1. They are easily caused mild common cold symptoms. I'm very sure many of us have experienced at least one in our lifetime having the common cold. At that time, nobody cared about the cause of this common cold is basically because of the coronavirus infection. Now, coronavirus is so famous because of the deadly disease, the COVID-19, due to infection of a new member of this family called coronavirus 2 or SARS-CoV-2. Other two coronavirus called SARS-CoV and MERS-CoV have caused pandemic in the past 2 decades, like in 2002 and 2003, the severe acute respiratory SARS-CoV caused a SARS epidemic that resulted in a 10% mortality. Similarly, the MERS-CoV caused the Middle East Respiratory Syndrome coronavirus, caused a devastating pandemic in 2012 with a 37% mortality rate. Now, let me give you some idea about the origin of the SARS-CoV-2. On the right-hand side, there is a cartoon demonstrate the natural reservoirs and the intermediate host and the target host in major coronavirus listed here. All coronavirus that have caused disease to human have had animal origins, generally either in bats or rodents. Then the question is, why do so many infectious diseases emerge from bats? There are over 130 different kinds of viruses found in bats. Meanwhile, you might be wondering, why are not the bats themselves affected by the virus? It seems that bats have developed special immune system to deal with it, and their system do not overreact to infection, which keeps them from falling ill from the many viruses they carry. And this shows that it is not always the virus itself, but the body response to the virus that can make us sick. In fact, what happens upon infection with the SARS-CoV-2, our immune system basically abnormally responded. As a result of that, we got like overreacted immune system. That we'll be talking about very soon. In the case of SARS-CoV and MERS-CoV, they were generally transmitted directly to the human from the camel, respectively, for the SARS-CoV and MERS-CoV. But for the 2019 SARS-CoV-2 was likely transmitted to the human through pangolins that are illegally sold in the Chinese market. Although I think many of you heard about that, there was some early speculation that SARS-CoV-2 emerged from a man-made manipulation of an existing coronavirus, but there is no evidence to support such theory. Now, let me share this slide about the transmission cycle of the SARS-CoV-2. The transmission cycle of the SARS-CoV-2 is showing the tentative transmission path from a natural host of bat to human, which is termed as zoonotic disease. Let me define what is a zoonotic disease. A disease that usually occurs among animals but can infect human in specific condition known as a zoonotic disease. They have largely affected the human population for the past 100 of years. However, with passing time, they have changed in several perspectives concerning their occurrence and pathogenicity, probably due to their capability of high frequency of mutation in their important protein like spike protein. So as for now, several proof indicates that CoV transmission occurs via zoonotic spillover, a term indicating the transmission of a pathogen from a vertebrate animal to a human host. Although the mechanism of such transmission is not very clear, therefore, it is a matter of concern. Certain factors determine zoonotic spillover, such as behavioral characteristics of the CoV and the susceptibility of a human host. Now, I think many of you know about the clinical presentation of the patients with COVID-19. Very common is the fever and headache. It's not very specific to COVID-19. Cough, it is also not very specific to COVID-19. The more specific of the clinical symptoms of the COVID-19 is the shortness of the breath in severe cases, pneumonia, of course, and the septic shock due to severe infection from SARS-CoV-2. Myalgia is also known as muscle pain and muscle ache due to coronavirus infection. Hemolysis is the coughing up of blood or blood-stained mucus. It's happened even like COPD or like pneumonia. So those are the some specific and non-specific clinical symptoms of patients with common cold as well as COVID-19. Now the question is, which organs are most severely damaged by SARS-CoV-2? And I'm very sure you all know this answer is that the lung are the primary site of injury by SARS-CoV-2 infection, which causes COVID-19. And the virus reaches the lung after entry in the nose or mouth. The reason is the receptor for SARS-CoV-2 robustly expressed in the epithelial cells of the lung, as well as renal epithelial cells like kidney cells, and of course intestinal epithelial cells and some other organ and their cells. It can infect entire respiratory system, all the way down to millions of tiny ear sacs called alveoli. And the difference of SARS-CoV-2 versus other SARS is that the other SARS virus generally infect the upper respiratory system, while SARS-CoV-2 infect the entire respiratory system, starting from, as I mentioned, trachea to all the way to the tiny alveoli. Most of the severe cases, the infection happen in these tiny ear sac called alveoli. The alveoli, literally, as you can see from here, keep us alive by bringing oxygen, as shown here, into the bloodstream and excreting the carbon dioxide, as shown here. But the SARS-CoV basically disrupt these processes. So really, this is a way the alveoli help us breathe. Due to SARS-CoV-2 infection, this exchange is disrupted, and the patient really suffered from is called the ARDS, called acute respiratory distress syndrome. Now, let me talk about the mechanism of SARS-CoV-2 infection. On the right-hand side, we have I have shown a pneumocytes, or you can say is the epithelial cells of the our respiratory system or lung, where the receptor protein called angiotensin converting enzyme 2 robustly expressed, where the SARS-CoV-2 virus can bind with a spike protein. And what happened next is that there's another molecule called TMPRSS2, which is a serine protease, does express by host, meaning our epithelial cells. It then bind and cleave the SCE2 receptor to activate it. In the process, the spike protein is then activated and cleave SCE2, and activated spike protein then facilitate this virus to enter inside the pneumocyte or the respiratory epithelial cells through a mechanism called endocytosis. And the genetic material of the virus, which is a 30 kb kilobase per single stranded plus RNA, is released as shown here directly into the cytoplasm. And then they hijack the host cells transcription and translation machineries or mechanism called the RTC, replication transcription complex, in a double membrane vesicles. And then they replicate and then they increase their own number inside the cells. And then slowly they get out of the one cells, and then basically they can infect more epithelial cells. So viral entry and the cell infection basically triggers the host immune response. And the inflammatory cascade is generally initiated, by antigen presenting cells, like dendritic cells, like macrophages as shown here, in a severe stage. Now, let me tell you about what happened upon triggering of the immune system. So inside the alveoli are immune cells called the macrophages, as I mentioned, and of course dendritic cells. They generally always keep monitoring if there is any invading foreign agent isn't there. And they generally act as an sentinels for the lungs. They recognize the danger and they try to get a read of it. In case they are not able to do it, then these macrophages attack the virus basically, and a battle that the immune system sometimes wins. If our body needs more help, it then recruit more immune cells or neutrophils or basically other lymphocytes. As shown here, once our macrophages recognize that there is a foreign body, for example, the SARS-CoV-2, then they communicate with the T cells. In our body, always there is T cells, which is in a resting conditions, when they communicate and then get activated, and then they expand their own population of different type of lymphocytes, like Th1 lymphocyte, Th2 lymphocyte. So Th1 lymphocyte basically responsible for production of several pro-inflammatory cytokines to induce the immune responses. On the other hand, the Th2 lymphocytes generally produces anti-inflammatory cytokines to counteract the immune responses of inflammation, otherwise inflammation going on. So that's basically happening in SARS-CoV infection in our respiratory epithelia. However, what happened actually under severe infection in our lung epithelia with the SARS-CoV infection, that the immune system goes haywire, results rapid release of large amount of cytokines, which is called the cytokine storm. It's huge uncontrolled amount of cytokines released inside the alveoli upon SARS-CoV infection in a severe disease condition. Here, I'm very sorry that you cannot probably see the real picture of different kind of cytokines increased in patients who are in ICU, meaning their situation is really, really severe, versus the healthy control and non-ICU patient, meaning their condition is not that severe, but mild. So these results actually reported in a great journal, probably you may know, the Lancet, where they have found that a number of pro-inflammatory cytokines and anti-inflammatory cytokines was increased in ICU patient and non-ICU patient compared to the healthy control. Let me mention a couple of them. For example, you can see the IL-4, right? IL-6 is a pro-inflammatory cytokine. It generally happens to rise upon infection, any kind of infection. And specifically, if you are aged, these IL-6 level really increase a lot in compared to the kids. So that's one of the major reasons why kids may not have that much severity of the disease compared to the aged person I'll be talking at the end. Several cytokines were listed here, as you can see, were higher compared to healthy control. So what happened basically in SARS-CoV-2, I think, is that in COVID-19, there is increased activity of the pro-inflammatory cytokines. As a result of that, there is an imbalance between pro-inflammatory cytokines and anti-inflammatory cytokines listed here. As you can see, these are the cytokines generally considered as the anti-inflammatory cytokines produced by T regulatory cells, and their job is to keep the immune homeostasis. Whereas the pro-inflammatory cytokines, or you may call it the pathogenic cytokines, produced by different kind of T lymphocyte subsets, as shown here, Th17 cells responsible for production of these pro-inflammatory cytokines. Th1, they are responsible for production of all these pro-inflammatory cytokines. And see here, because of this imbalance between these two cytokines, pro-inflammatory and anti-inflammatory, probably leading to the abnormal immune response or overreaction of our immune system, leading to them to I think septic shock and damage of other organs. In typical cases, one would expect the situation like this. There is a balance between the pro-inflammatory response and anti-inflammatory response to resolve the infection as a result of tissue recovery, which is not happening in SARS-CoV-2 infection. In SARS-CoV-2 infection, as I mentioned, there's an imbalance. The pro-inflammatory cytokines is higher compared to anti-inflammatory cytokines level. Now, what is the impact of the imbalance between pro-inflammatory cytokines and anti-inflammatory cytokines on the flooding of the alveoli that cause too hard to COVID-19 patient to breathe? So that we'll be talking about, possibly because the disrupt, meaning the cytokines, disrupt probably the transport protein that is equipped in alveoli epithelial cells responsible for to make this alveoli clean, clear, and very little amount of fluid. That I'll be talking to. So two things here is that the response from the SARS-CoV-2 virus and the response from our immune system, overreaction or explosive responses make this coronavirus really so deadly. So in the worst case scenario, what happened is that probably the wall of the alveoli being to break down. As a result of that, what happened, the fluid generally rushes from the blood vessels into the alveoli as shown here, filling them up and blocking the exchange of gas because the alveoli is full of fluid. So there is very little space for the gas exchange. So that actually make the COVID-19 patient hard to breathe. So when this happens, we cannot excrete enough carbon dioxide from the alveoli to the blood or nor absorb enough oxygen. So it becomes much more difficult to breathe. And then the obvious question is how and why this fluid accumulation does happen. As I mentioned, the primary respiratory unit or alveoli are composed of a single polarized epithelial cell layer as shown here. As you can see here, those are the polarized epithelial cells lined in the alveoli. And that basically separates the gas-filled compartment. So this is always the gas-filled compartment and the pulmonary circulation, blood, right? One of the main functions of the alveolar epithelial cell is to control the ionic composition and the volume of the fluid. So volume of the fluid should be very thin to hydrate this the inside of this alveoli, at the surface of this alveoli. And two predominant cell type has been identified. They are the type 1 is called AT1 and the type 282. And both cells type contribute to alveolar fluid transport by the help of the transport protein as shown here, mainly the sodium channel and another one, the chloride transporting channel called CFTR. Probably many of you heard about this channel protein, which causes diarrhea in our continent. And recently what we identified is there is another chloride channel called is TMEM16A, or calcium activated chloride channel. Probably that channel may have some role in the induction of the fluid accumulation due to SARS-CoV-2 infection, and that currently we're working on whether we can inhibit this one of this channel, can rescue or can remove the fluid accumulation that helps to breathe by SARS-CoV-2 infection. So maintaining the fluid layer in the alveoli, basically, as I mentioned, critically depend on sodium absorption mediated by the sodium channel called ENaC and the CFTR chloride channel, and perhaps the new channel, which people do not know, probably whether it has a role or not, probably the TMEM16A. So what happened during the SARS-CoV infection is that, as I mentioned, that there is a cytokine storm happened due to SARS-CoV-2 infection. And those are the pro-inflammatory cytokines really increase in our circulation, more specifically inside the alveoli, and probably they inhibit the transport mechanism here as shown here. And then probably because of that, it may have other effect too. It can damage the lining of this epithelia, and as a result of that, the fluid can get into it. So that's probably one of the major reasons why the alveoli get flooded due to SARS-CoV infection. Now let me talk about, or probably you also know why children have reduced susceptibility to COVID-19. I did some research, and what I have found, there are 4 clues that could explain why children have reduced susceptibility to COVID-19. Most of the COVID-19 patients are like very aged people. One clue is probably that the coronavirus associated with common colds, which more frequently happen in children, which may offer some protection because already because of their common cold, there are some like cytokines already is in there. And probably that can give some protection to them not to have the SARS-CoV infection. And it is known, or people have shown before, that 2 important factors that probably facilitate the entry of the SARS-CoV-2 called TMPRSS2, which is the protease and receptor, their expression is very low in the nasal mucosa of the children. So this is the 2 main clue probably and can explain why children have reduced susceptibility to COVID-19. And other one is that those are the anti-inflammatory cytokines that probably already elevated in their alveoli, in their circulation upon common cold virus infection. Therefore, the children can tackle or can limit the infection of SARS-CoV-2. That's the 3rd clue. And another one is, as I mentioned earlier too, that children produces lower level of inflammatory cytokines, which is the IL-6, which is highly increased due to SARS-CoV infection in aged people compared to the children. So these are the 4 clues probably can explain why children have reduced susceptibility to COVID-19. And then another question probably you may know or you may heard, why are more men dying from COVID-19? Though nobody can yet explain the oddity, researchers are hot on these issues. Um, the novel SARS-CoV-2 tends to affect men more severely than it does women. The reason is that the sex hormone, estrogen, and the testosterone plays probably not yet evident, plays a role according to the previous research on respiratory illness. Or perhaps it is because the X chromosome, which women have two of, but men have only one. And these X chromosome has a large number of immune-related genes, which gives women a more robust immune system to fight off the coronavirus SARS-CoV-2 compared to men, because men have only one X and one Y chromosome. And probably the virus is hiding in the testes. The reason is in the testicular cells, there is an abundant expression of its receptor SE2. Probably the protal of the high expression of the SE2 allows SARS-CoV-2 into the cells. So these four clues can probably explain why are more men dying from COVID-19. Now, let me share some take-home message before my final. I have couple of more slides to end. So COVID-19 patients have significant pulmonary inflammation due to the overreaction of our immune system, leading to the flooding of the pulmonary alveoli, because those cytokines probably affect the alveoli ion transport system, leading to the flooding of the pulmonary alveoli. And these prevent the normal gas exchange between carbon dioxide and oxygen. As a result, there is a hypoxemia and then causes mortality. The direct attack on other organs by disseminated SARS-CoV-2, the immune pathogenesis caused by the systemic cytokine storm, and the microcirculation dysfunction together lead to viral sepsis and then death. Therefore, effective antiviral therapy and measures to modulate the alveoli ion transport system. For example, if we can find an inhibitor that could inhibit or modulate the sodium transport and inhibit the chloride secretion, inhibit the fluid accumulation in the alveoli, along with the modulation of the innate and adaptive immune responses, would help for breaking the viscous cycle, and that would improve the outcome of the patients. Now, I have two more slides I want to share, but let me give you the concluding remarks that considering the recent outbreaks of COVID-19 caused by the newly emerged coronavirus SARS-CoV-2 and the potential of future recurrence of SARS, development of effective and safe vaccines against SARS-CoV-2 remains a high priority. Now, I know many of you are very much eager to know what happened to vaccine, even though if there is a vaccine, really going to be effective. So vaccine against COVID-19 is a hope or a hype. Let me bring one slide that can give you some information about, or it is self-explanatory. You can understand really vaccine is a hope against the SARS-CoV-19, or we have to live with it. So definitely vaccines are one of the monumental achievements in human medical intervention to mitigate the dispersion and impact of the infectious disease. So here, I'm going to share one thing about the timeline for the development of various vaccines including polio, Ebola, SARS-CoV-1, MERS, and SARS-CoV-2. If you look at the polio, and here you can see since the outbreak of this disease, it took 60 years to have a protective vaccine, right? Let's talk about the Ebola. Since it started, our outbreak happened in 1976, and then the Ebola genome sequence in 1993. Then the Canadian team took almost 15 years to have a vaccine against the Ebola. How about the SARS-CoV-1? Outbreak started in 2003 and genome sequence in 2003, right? Fast clinical trial also happened. What happened? No vaccine in 17 years against SARS-CoV-1. What about MERS? So it was like outbreak started or pandemic started in 2012, and the MERS genome sequence in 2013. Several approaches, were taken to have a vaccine against MERS. No vaccines since its outbreak, almost 6 years. SARS-CoV-2 outbreak happened in 2020. Nobody knows whether really a vaccine is going to be available in, let's say, 2 years, 3 years. Even though available, whether we do not know going to protect us. You know about the influenza vaccine, right? You have to take every year because it does not give us long-term protection against this infection. So this timeline, I'm very sure going to tell you, probably there is a hope or maybe hype. We have to live with it. Because if you look at the history of the vaccine against virus, HIV, there is no vaccine available for the HIV virus. And look at other disease, diarrheal disease in our continent. There is no effective vaccine available against the Vibrio cholerae. Is a probably hope or hype? I do not know. But as we wait for scientists and healthcare professionals to develop a vaccine for COVID-19, what we can do? We have some responsibility probably. And the responsibility, I would say, is 3 W's. What are they? Wait 6 feet apart, maintaining social distancing as shown here. Wear a mask, cover your nose and mouth. Wash your hand. Definitely these 3 approaches could help us to limit the infection, to spread the SARS-CoV-2 infection. Now, washing of the hand is very important. How you're going to do that? Probably, I don't know whether this video is going to help, but at least if you see the sequence of the washing of the hand is very important. Just a simple soap can help us to kill the virus. Now, another thing is social distancing is very important. Let me bring one math behind the social distancing that can help to reduce the infection. Here, someone did some math. If we do not maintain the no social distancing practice, then what happened in 5 days period, one person can infect 2.5 number of people or others. In 30 days, that can spread to 406 people. If we reduce the 50% reduction in the social exposure that's happening in many Asian countries, what happened then? One person infects probably 1.25 people, almost like 50% reduction. That could help to reduce infection to 15 people. If you reduce the 60% social exposure, or that could help also, like in a further as shown here. So social distancing really helps to reduce the burden of this disease, and we need to maintain it. So this is all about giving some pathophysiological aspect of the disease, COVID-19, and of course our own responsibility before available of a of a safe and protective vaccine, we can help to limit the spread of this deadly disease. And finally, I thank you all. Now, probably, if you have any question, I will try my level best to answer it. Thank you for listening.
Thanks a lot, sir. I think the talk was very informative and it was really interesting. A lot of complex things were presented in a very simple manner, and we have got many questions, so we would like to take them one by one. And the first question is more of a kind of information that they want to ask. It's been asked by Mr. Asim Azhar, and so he wants to know that, as you said, that there are more than 130 odd viruses in bats. So, can you suggest for any reference of where to, look out for them, or for as a repertoire of all those viruses?
Yeah, I think if you in the PubMed probably you can get it. Yes. Specifically, if you want that references, I can send it to you after the talk. I don't have on hand right now.
Okay. Then I think this is another question is which is about. Okay, this is more of a technical question, but since he has asked, so I'll just put forward. Which software have you used for the presentation?
Oh, it is a PowerPoint presentation, right? So I don't think so. It is a very sophisticated any software I use. It's a very normal one.
Exactly. Okay, so now from your talk, Mr. Ankur wants to know that what is your idea or about your understanding about how did the virus get transmitted from bat to humans, by food or some other way?
Yeah, that's nobody knows, but speculation is probably somehow it get into the human through a market, right? The one market where most of the meat, all those is sell. Probably one of the possibility. Nobody knows yet. Or maybe it's probably continuous environmental pressure because of the bad number is decreasing, but virus needs to survive, so they're looking for the host that they can live with for longer time. Or I think this is because of the survival for them.
Then Miss Meera wants to know what is MODS?
What is that?
MODS, MODS. I think multiple organ. I think that is you have used in the presentation. Yeah.
Yeah. So what happened actually, because the receptor for the SARS-2 does express in many organs, including the renal epithelial cells, gastrointestinal epithelial cells. That's why many people infected with SARS-CoV-2 may have diarrhea, renal failure. Renal failure, I mean to say, is when your kidney does not work. Definitely, the waste product cannot get out of your body. So definitely that can then increase like the uric acid level, urea level, creatinine level going up. And that's because the receptor ACE2 does robustly express all other organs. That's giving them opportunity to infect other organs. But people are still trying to find out what is the pathophysiological alteration happen due to infection in the renal epithelial cells. Even my own area of expertise is the gastrointestinal epithelial cells. Why some COVID-19 patients suffering from diarrhea? Though not very common, but in a few percentage, at least 5% COVID-19 patients have like severe diarrhea, CKD diarrhea. So probably the reason is the receptor expression.
Thank you, sir. Ms. Meera has another question that if you could explain a little bit more about ENAC1.
Yeah, so ENAC is the sodium channel, and the job of this protein is to absorb sodium. And the other channel, the called CFTR, responsible for the secretion of the chloride. And there is a balance between the absorption and secretion always happen in our secretory epithelia, including intestinal epithelial cells, renal epithelial cells, and of course the respiratory epithelial cells, to maintain the hydration of the intestinal lumen or the alveoli, so that alveoli can function properly, or if there is a dysfunction happen due to infection, if there's inhibition of the sodium absorption, that means the chloride secretion probably accelerated, and probably there is a osmotic gradient happen between lumen in the case of intestine, or alveoli in the case of respiratory epithelia, in respect to the outside of the epithelial cells. That bring the fluid in to the lumen or alveoli. So that's the way the transport works. So transport have to be very fine tuning between absorption and secretion to maintain very thin layer. If that is being disrupted, then other, meaning, flooding is going to happen. That's like in diarrhea, like as I said, the edema in alveoli.
Thank you, sir. Mr. Upal Das Ghosh has a question that if after recovery from COVID infection, whether it is possible that the fluid accumulation in alveoli will still continue and that results in the recurrence of breathing problem.
So the fluid in alveoli that I described is in a severe condition where probably you may not have that big chance to come back. Probably you are going out of business, probably leading to death. If you recover, if there is the ability to, to reduce the fluid accumulation, that's the whole reason, we're trying to find out whether any inhibitor that could help inhibit the fluid accumulation that we propose to the NIH, let's see whether we can get the money or not, would help, definitely. But nobody actually looked in that way.
So Mr. Shakilur Rahman from IIT Kharagpur, he wants to know whether the inactivation of X chromosome, or that is the bar-body formation, what role does it have? Does it play in this infection or anyhow? Because you talked about gender.
Yeah, it is under investigation. Nobody knows. But the explanation I gave you is a simple explanation with a possibility that because the X chromosome responsible for many immune system related genes may help to fighting of the disease, but nobody knows yet.
Ms. Shraddha Mahendra, she wants to know that what extra precautions should be taken by cancer and HIV patients since they are already immunocompromised?
Yeah, that's a hard question and not my expertise. Doctors and clinician, who does research probably can answer that question. I do not have the expertise and I don't know anything about it. Okay? I don't know whether in the audience there is any doctor. They can tell us something, but I do not know. Sorry about that.
Mr. Sudeep Som wants to know why SARS-CoV-2 is known as novel coronavirus.
What is that?
Why SARS-CoV-2 has been called as novel coronavirus?
Oh, because so as I mentioned earlier that the other two coronavirus, SARS and MERS, right? They have named like the SARS, I think the SARS-CoV-1, and MERS as itself as a MERS, and this is a novel one because there is a identity, 75% identity, I guess, with MERS, and 50% identity with the SARS-CoV-1. With the SARS-CoV-2. So there is a big differences in their genetic material, meaning there is a big differences of SARS-CoV-2 with other two SARS-CoV-1 and MERS. Both three are responsible for epidemic and the pandemic, but there is a big difference of their genetic materials that make this SARS-CoV, SARS-CoV-2 as a novel, because there is a big differences in the genetic material compared to other two, MERS and SARS-CoV-1. That's why.
So there's another question by Mr. Rahman. This is related to a vaccine. Now, what he says is that if the spike protein of SARS-CoV-2 is very prone to mutation, then is it possible that even if we get a vaccine which is made against COVID-19 strain from India, but it will not be effective against the other strains from US, Europe,
Yeah,
and the
that's
different
true.
strains.
That's true. That's why, the last slide, I just put in last night, while I was working on this presentation, that, yeah, it's a good one, to share with them. Is a hope or hype? That's why I put all the history. Most likely, as you can see, 17 years too, right, for a, for other vaccine against the virus. Looks, HIV, there is no stable vaccine available. No safe and, a long time that could actually give us long time protection. So, it's very hard, very hard. Probably that's my own opinion. Probably may not have.
Only time will tell.
Yeah, time will tell. But mostly you have to take probably every year. Like the influenza virus. Like every year we have to take a vaccine against the influenza.
Okay. Now this is again question another. I think this is related to the X chromosome inactivation that was before. This has been asked by Ms. Taslima, and she wants to know whether there is a possible role for the X chromosome inactivation in the female body related to the COVID-19 genome in the cell. So does it have an impact if the genome is present in the female cell? Does it have any impact on the X chromosome inactivation or any correlation between them?
Yeah, definitely there is a correlation. That's why men are getting more infection than the women. But in the context of uh, in the context of in the therapy, I do not know, whether inactivation could help. And it may have some other effect. I do not know.
Fair enough, sir. There's another question by Dr. Madhusar, who is one of our colleagues at Alia University, and he wants your opinion about how competitive or effective would be the herd immunity against SARS-CoV-2. A lot of is being said about it, but what is your opinion about it? Herd immunity.
It's hard to say, It's hard to say. It's probably the people who are really clinician, who are doing research in the context of immunology. Probably they can tell. I do not have any information, any idea, and I should not make any comments on it. Okay. I'm sorry, I do not know that.
Yeah. There's, I think, another question, not really a question, but I don't know whether if you would like to comment on it. I'll just put it forward for you. In your idea, whether it is a natural virus or whether it is a manipulated one?
Definitely, it is not the manipulated virus. Definitely, because I keep follow all the report. For example, a group of scientists from USA, they visited there, and they published a report. So they are a member of the National Academy of Science, USA. Probably you cannot ignore their report, And they have clearly mentioned it is not a man-made virus. They have a very good viral laboratory in China, and the big people who has who are very famous virologists, and they already investigated that, and they clearly mentioned it is not a man-made virus. It is a natural virus. But it is being politicized by by politicians across the world. It is not a man-made virus.
Fair enough. There is another question by Miss Meera, and she wants to know if you can explain a little bit more about the interleukins regulation and their impact with reference to COVID.
I didn't get the question.
If you could please explain a little bit more about regulation of interleukins and their impact with reference to COVID.
Yeah. So that's we are also working on. We know that when there is infection happen, always there is a rise of cytokines. Mostly first in the first few hours of the infection, the pro-inflammatory cytokine is released. That facilitates to go the inflammation, to initiate or facilitate the inflammation. Once the inflammation happens, basically by the influence of the pro-inflammatory cytokines, then they can um, act on some other immune cells, for example, regulatory immune cells, then that helps to produce more anti-inflammatory cytokines to counteract the pro-inflammatory cytokines, to have a very nice balance between the pro and anti-inflammatory cytokines. Until the body, our immune system can maintain that balance, then you are fine, you're good. You can recover from the disease, you can resolve the inflammation or infection. But for the SARS-CoV-2, there are a couple of factors, still people are working on it. That the two factors, one factor is from the virus itself, and other factor from the host. Probably somehow overreacting our immune system that lead to imbalance between the pro-inflammatory cytokines versus the anti-inflammatory cytokines. Results that pro-inflammatory cytokines is so high, so heavy compared to anti-inflammatory cytokines that are not able to resolve the inflammation or infection. So inflammation going on, not checked, not counteracted by the anti-inflammatory cytokines. As a result of that, you know, we are having this issue, or can affect so many organs other than the lung, and finally it's leading to the that is called multi-organ failure. So basically, you can remember there is imbalance. And that imbalance happened because of the overreaction of our immune system by the action of both the coronavirus itself and the human host immune system. Still, people don't know, but definitely there is a cytokine storm happen upon infection in a mild to severe cases.
Fair enough. So there's just one last question, and which is again more of an interjection from your side. That what according to you are the recurrence chances of infection in a recovered person? This has been asked by Mr.
Yeah,
Rashad.
this is happening. Now even like adult, who has a COVID-19 like SARS-CoV infection and recovered, and then getting the infection again. So there is a possibility. There is a chance. It all depends upon, again, your immune background, your immune system background. If your immune system probably suppressed, or you're taking some medicine that could, for example, in a steroid, the people who are taking steroid probably they have a much more chances to have second infection because their immune system is suppressed by the action of the steroid. So probably, people are not recommending to have a, taking steroid. So probably, yes, there is a chance, and it depends on many factors, your own immune background and kind of medicine you're taking. So many things you need to consider.
Thanks a lot, sir, for taking all the questions and explaining it in a very simplified manner. There is, sir, one request from multiple participants. I am just putting it forward. I don't know whether it is feasible at your end or not. A lot of participants are requesting if you can share the PPT. So it's totally
sure, why not?
Okay.
Yeah, I can send it. I can send it to you, and then I don't know how to share with them.
We will forward it to the registered participants because we have their mail IDs. So
okay, sure, no problem.
I think so that answers the queries by all the participants who are asking for the PPTs. Sure. You will be getting it along with the feedback. Thanks a lot, sir. Thanks a lot.
No problem. Thank you very much. You're welcome.
Yeah, I would now like to call upon Dr. Mudassirul Huda to please give the vote of thanks.
So thank you, Dr. Savda. And first of all, gratitude is one of the virtues of the educated. So on behalf of the Department of Biological Sciences and Alia University, I take this opportunity to first and foremost thank our guest, Dr. Kazi Mirajul Haque from University of Maryland, for accepting our invitation and enlightening us with a fruitful and thought-provoking session. We are hopeful that it would add to our research endeavors in future. I would also like to thank our Vice Chancellor, Professor Mohammad Ali, Registrar Dr. Said Nur Salam, and Dean Faculty of Science and Technology, Professor Mehdi Kalam, for their constant support and encouragement for all the endeavors that we have pursued till date, and we are equally hopeful that they will continue to do the same in future. It is our privilege to have Professor Biswanath Chakrabarti as our mentor and guardian of the department. His critical inputs on various occasions has helped us in bringing all our resources together that led to a successful culmination of the department's first online seminar entitled Bio-Illumination 2020. I would like to extend my sincere thanks to Dr. Masroor Alam, Department Head of the Department of Biological Sciences, Aliyah University, and Chairperson of Bio-Illumination 2020, for his constant support and backing of the whole team. I also thank Dr. Sabda Ali, convener of the program, for conceptualizing and putting forth an extraordinary effort to make this event come into existence. A special thanks to Dr. Mehboob Haque for his consistent efforts in taking care of the registrations, feedback forms, and answering queries of the participants with regards to the program since its inception. It was indeed a tedious job, and our applauses to him for an efficient work. I also thank my colleagues Dr. Zakir Hussain, Dr. Kavita Sheikh, and Dr. Muhammad Abu Imam Sadi for the vital contributions at various stages of at the front end and the back end of the management that goes into preparation of the webinar. Lastly, I would like to congratulate all the participants for being a wonderful audience. We would hope that we all have gained something from today's session. Thank you once again on behalf of the Department of Biological Science, Ali University. Over to you, Dr. Sab.
Thank you, Dr. Huda. We would now request all the participants